الآن مع عرض عن مسير المؤتمر خلال السنوات السابقة. حضرة رئيس الجامعة اللبنانية البروفيسور بسام بدران المحترم، مؤسس مؤتمر بيروت وراعي هذا المؤتمر بنسخته العاشرة. حضرات العمداء ومدراء الكليات المحترمين، حضرات الأساتذة والموظفين والطلاب المحترمين، حضرات السادة الكرام الداعمين لهذا المؤتمر والمشاركين في تنظيمه، أيها الحضور الكريم، مهما طال الليل فالفجر آت. ها هي الجامعة اللبنانية التي رغم الصعاب التي تعصف بها تبقى متسلحة بالأمل لتدعم جسور المعرفة مع المجتمعات العلمية العالمية، فتطلق اليوم المؤتمر العلمي العالمي بيروت بنسخته العاشرة حول علم الأورام والأحياء السرطاني وعلم المناعة. Ladies and gentlemen, dear colleagues, after these many years of darkness due to COVID-19 pandemic, economic and financial crisis, the sun rises and the Lebanese University keep her role of spreading culture and building bridges with international scientific societies. Today, the Lebanese University launched the Biobeirut International Meeting in its 10th edition. أهلا بكم في رحاب جامعتنا الوطنية، والكلمة الآن إلى رئيسها البروفيسور بسام بدران.
صباح الخير، صباح الخير جميعا. طبعا اليوم بعد شغل كبير من قبل اللجنة اللي عم تشتغل على تنظيم المؤتمر، بعد شغل كتير كبير من الشركاء اللي كانوا داعمين اساسيات لهالمؤتمر، كان شركة بايوتك او شركة إدارة او انستيتيو فرنسي أو بازلكي. قدرنا في الظروف الصعبة والوضع الاقتصادي الصعب نرجع نعيد الحياة للباحثين وللطلاب اللي راغبين أول حرب. طبعاً ظروف صعبة، ومر بالبلد بظروف كتير صعبة، الجامعة بظروف اقتصادية كتير وإدارية كتير صعبة. إن شاء الله بالتعاون بالأمل نقدر نرجع نستعيد مكانتنا، نقدر نرجع نرد الروح للمؤتمرات العلمية اللي كانت تصير بالجامعة، نقدر نرجع نسوا الطلاب والأساتذة ليبعدوا شوي عن الهم المعيشي اللي هو هم كتير كبير، بس نحاول سوا نرد الأمل لمستقبل أفضل بالجامعة وللطلاب والأساتذة. كلمة صغيرة بس للضيوف اللي جايين من برا. First, I would like to welcome all of our guests that are coming from outside of Lebanon, from Italy, from France, from USA, I don't know if here, Habiba, and welcome again in Biobeirut 10. I'm really happy and delighted to welcome you, all our dear guests, and all our invited professors and our students in this version of Biobeirut. Despite the financial and commercial crisis in Lebanon and the Lebanese University, we'll still be able to organize this Biobeirut meeting. A big thanks for our supporters, either Biotech, Etico, and Institut Français and Pacific, for the effort that they did. Aussi, je souhaite remercier énormément l'Institut Français pour la contribution directe à cette congrès. Je souhaite aussi remercier tous nos invités pour tout leur effort et surtout Dr. Walid Rashid. Why? Because when we have discussed do this meeting, the situation was really critical, and from financial point of view, it was almost impossible. Walid told me, look, Bassam, we should restart back. We started this meeting more than 10 years ago. It's the 10th version. He told me, look, don't worry. For the airline tickets, we will cover, those are coming from, we will cover directly. Just find the money for all the rest of the people. Here, Pierre Saloum, and Mr. Fawaz, Mr. Ali Nasser did a great job, because if they don't support us, we'll not be able to do it. And Institut Français come to add with validity the final version, the final tuning that allow us to be able to do it today. Welcome in Biobeirut, and I will be happy to welcome you, and we will try to have a very nice meeting and share all this information with all of you. Thank you very much.
So now we will start our first session with Professor Philippe Benaché. I will invite the moderators, Dr. Hassan, she will arrive, Dr. Kurdi, Dr. Mazen Kurdi, Dr. Eva Ahmed.
Good morning, everyone. We are pleased to start our first session with Dr. Philippe Benaché with the title Extracellular Vesicle for Cardiac Repair. Please, Dr. Philippe Benaché is a clinical cardiac surgeon at the Hospital Hôpital Européen Georges Pompidou, professor of thoracic and cardiovascular surgery at the University of Paris, and co-leader of an INSERM team devoted to cell therapy and cardiovascular diseases. Dr. Philippe, we are happy to have you with us today, and the floor is yours. Thank you.
Well, thank you very much. And obviously, my first words will be to thank you for the privilege, the honor, and the pleasure of this invitation, and of being here today with you. In your country, you know that there is a special relationship between Lebanon and France, and I'm especially happy to be here today. So, let's start by the rationale for using extracellular vesicles for cardiac repair, and let's try to make it as simple as possible. When cardiac cell therapy started, the initial hope, which turned out to be wishful thinking, was that once the cells better like that? Yeah. but you listen to the introduction, so I don't have to come back. So when we started cardiac cell therapy, the initial hope was that once transplanted in the heart, the cells would make, say, a new myocardial tissue. And as I just mentioned, that was wishful thinking. Reason being that no matter what you do, the transplanted cells ultimately will die. And they die for a variety of reasons, which I will not detail. But at the end of the day, they die. Now, the puzzling observation is that although the cells have died, you still may have a functional benefit. So people try to figure out why. What was the mechanism behind that? And this led to the so-called paracrine diseases, whereby cells, although they die and before they die, release a blend of biomolecules, many of them being packaged in extracellular vesicles. And these vesicles, these biomolecules, will activate endogenous reaper pathways. So the reaper will come from the heart itself after some mechanisms have been triggered by the biomolecule released by the cells. Now, the initial experiment that we did, as many others, was very simple actually. That was the time I was transplanting surgically embryonic stem cell derived cardiovascular progenitor cells, SSA1 positive cells. And what Nisa, who was a postdoc in the lab at that time, did was very simple. She took the cells, exactly the ones that I was transplanting clinically, cultured them for 48 hours, and then split the batch in two components: the cells and the extracellular vesicles from the same batch of cells. And then we used a standard mouse model of chronic heart failure, and in a blinded fashion, either transplanted the cells or injected the EVs from the same cells. And we took left ventricular and systolic volume as the primary endpoint. And you know that the lower the volume, the better the prognosis. Well, exactly similar outcomes. Exactly similar outcomes, no matter you transplant the cells or you inject the secretome. And when we looked at the pathways that had been turned on in the recipient hearts, basically similar pathways had been turned on. And since then, there has been compelling evidence across a wide variety of preclinical models that the effects of the cells can be duplicated by administration of their secretome. So just a few words about the extracellular vesicles. These are nanoparticles. I will not go into too many details, but basically they're key drivers of intercellular communication. They have a very rich payload that they can shuttle into recipient cells. And I just wanted to show you a video. Just look at the arrows. The arrows indicate the extracellular vesicles, and these are cardiac cells in culture. And if you follow the arrows, you will definitely see the vesicles being internalized into the recipient cells. So look at the arrow, and you will see the vesicle getting inside the cells. So this is not fancy. This is something that really happens. Now the question is, once the vesicles have been internalized in the cells, they release their payload, and this is the mechanism for activating the endogenous reaper pathways I was talking about. And it seems that one of the major mechanisms of action is really a reduction in inflammation and fibrosis, which are hallmarks of heart failure. And just as an example, this is once again a very simple setup with injections of either EV, extracellular vesicles, or cardiovascular progenitor cells. And long story short, if you look at the main results, you see that in the myocardial tissue, you have a reduction of pro-inflammatory cells, both with the cells and with their EVs. So if you admit that there is equivalence, then the question is, why do you use the EVs instead of the cells? Well, there are different reasons. Some are good reasons, others are poor reasons. First reason is that it's easier to manufacture EVs compared with cells. Wrong. If you need and if you want to have good, well-functioning EVs, you have to start from good cells, no matter. So this is not the good reason. Second reason, it's easier from a regulatory standpoint. Wrong. Absolutely wrong. These EVs are not classified as AT&Ts, but biological medications. The regulatory path is slightly different, but it's not easy. More accurate dosing, probably yes, because it's a suspension. It's a non-Newtonian fluid, so you probably have a more even distribution than when you inject cells. If you do multiple injections of cells, the cells tend to sediment over time, and you end up with an uneven distribution of the cells. It's probably easier to have more accurate dosing with a non-Newtonian fluid, and I will show you an example in a few minutes. Better potency assays to test the bioactivity, definitely yes. It's easier with a solution of EVs than with cells. I just show you one example. You take cells, you stress them with a pro-apoptotic agent like staurosporin, and this is the blue, the light blue here histogram, and then you expose the same cells with EVs and look at whether you have an improvement in cell viability. It's a very straightforward assay, and it allows very simple dose-effect relationships, probably easier than with cells. Maintenance of bioactivity is important because they can be used as an off-the-shelf product, but probably the most clinically significant advantage is the lack of immunogenicity. If you take embryonic stem cells or even iPS or allogeneic cells, if you transplant them, you will trigger an immune response. This is not the case with EVs. This is the immune profile of the cardiovascular progenitor cells that we have been using and that we will use clinically in the next weeks or so now, since we get approval for the clinical trial. And you see that there are some markers which are expressed. Conversely, if you look at the EVs from the same cells, they really look immunologically neutral, and actually they do not trigger CD4 or CD8 proliferation in contrast to the controls, and likewise, they do not cause NK granulations. So this is really the greatest advantage. You do not need to give immunosuppression to the patients. Now, what are the issues you have to address if you want to move to the clinical trial? Well, the first is the choice of the parent cells. And we now have good evidence that not all cells are equal, and that they should comply with two major requirements. First, the cell should be as close phenotypically as possible to the brain. That is starting from brain-committed cells, and so on. And number two, cells should be young at an earlier stage of differentiation, simply because the earlier the cell, the richer the secretome. So the result is that it qualifies cardiovascular progenitor cells as ideal candidates, although mesenchymal stromal cells have also been shown to be effective. Nevertheless, this is an interesting comparison between mesenchymal stromal cell and cardiac stem cells. If you look at a fairly crude endpoint, like ejection fraction, basically they look similar. If you go a little bit further further and look at more subtle indices of cardiac function, like left ventricular volumes or wall thickness, the picture is slightly different, and you see that consistently the cardiac stem cells outperform the mesenchymal stem cells. These are left ventricular and diastolic volume, and this is wall thickness. And this makes sense because the cargo of the cell differs from one cell type to the other. And basically, the cargo of cardiac committed cells is better tailored for cardiac repair. So for this reason, we started from IPS, then generated cardiovascular progenitor cells, took the vesicles, tested them in the mouse model of chronic heart failure, and as you can see once again, the EVs are doing as well as the parental cells. They even slightly outperform the parental cells. And one likely reason for that is that, back to what I was saying about non-Newton fluid, the likely reason is that administration is more uniform, is more even with a liquid solution than with cell suspension. If you take left ventricular and systolic volume once again as the primary endpoint and stratify the animals between responders and non-responders, you see that with the EVs, the values are much less scattered than they are with cells. This is probably why they're doing better. Once again, looking at the pathways, you find activation of mechanisms which overall contribute to cardiac repair. So just a few words about manufacturing, because for a while it was considered very difficult to manufacture EVs in a clinical perspective. Now I'm a clinical surgeon, so I'm interested in clinical applications, and I just want to illustrate by a couple of slides the process that we have followed to move to the clinical trial, which will start now, as I told you, in a few weeks. In collaboration with a US company, the first step is the manufacturing of IPS, and they have a special process for reprogramming somatic cells into induced pluripotent stem cells. Then the IPS have been differentiated into cardiovascular progenitor cells. And as you can see, 2 banks have been manufactured. So these are clinical grade cardiovascular progenitor cells. Then the cells, the progenitors, have been frozen and shipped across the Atlantic to our GMP facility in Paris. And then started a downstream process which comprises 3 main steps. The first one has been the thawing of the frozen progenitors, just a few days of scale-up, and then obviously the collection of the conditioned medium. The second step has been the filtration and purification of the medium, which is being done by tangential flow filtration. And here is something interesting. The issue is not so much to choose the technique. The only technique that you can use for clinical purposes is tangential flow filtration. The issue is the degree of purification. And for a while, people thought that we should strive to highly purify the medium. Well, there is the possibility of controlling the size of the pores of the membrane. And depending on the size, you can retain or discard whatever you want. Well, there is increasing evidence that it's not necessarily very good to try to highly purify the medium, and that, so to speak, the soup is what works. That is, most of the secret of the extracellular vesicles, but also soluble factors, and so on. Let me just show you one example of that. This is an interesting head-to-head comparison between exosome fraction, extracellular vesicle, or the protein fraction.
And you see that in vitro, basically, they perform in a very similar fashion. Now, in vivo now, it's interesting to see that once again you find the equivalence between the cells and the exofraction. You find again the equivalence between the exo and the protein fractions, but the better outcomes are seen when you mix all the fractions together. And I just want to show you one example outside of the heart, which basically shows the same. This is, a completely different setup with a comparison of the conditioned medium, the whole bulk, versus purified EVs. And if you look at the red arrows, you will see that regardless of the endpoint, you consistently find similar outcomes with a whole medium versus the fractionated EVs. Similar outcomes. And the final stage is being the fill and finish and the freezing of the EVs. I will finish now with a few words about delivery. Delivery is critical in the clinics, obviously. You have very nice EVs in your dish, but if you cannot transfer them, it will not have any therapeutic value. If there is a surgical indication, which I'm interested in, then there is evidence that it's probably good to incorporate the EVs in slow-release polymer. It's probably better than the snapshot injection release polymer. And I'll just show you one example. Here's a study we recently completed with either EVs or EVs incorporated in hyaluronic acid. And the data, I think, speak for themselves. You get better outcomes if the EVs are incorporated in the polymer rather than just injected one shot. Now, what is interesting is that many of these patients do not require open heart surgery. And so, what is interesting to consider is the intravenous approach. The intravenous approach has a lot of advantage that I will not detail, but initially it looks completely counterintuitive. Completely. Why? Because if you look at biodistribution patterns, when you inject cells or EVs intravenously, they end up in the spleen, the liver, the lungs, but very few, if ever any, in the heart. Nevertheless, if you look at experimental studies, inject intravenously either EVs or cells, they have cardioprotective effects. So once again, it's different to reconcile the fact that they don't reach the heart, and nevertheless, they seem to have cardioprotective effects. And we confirmed that recently in two different models. This is a chemotherapy-induced model of cardiomyopathy, where IV injections of EVs definitely improve strain by MRI, which is probably the most sensitive index for cardiac function. And we confirmed that in a rat model, also of doxorubicin-induced cardiomyopathy with repeated IV injections. So this has led now to what is called bioreactor hypothesis, whereby the cells or the EVs, once trapped remotely in spleen, liver, lung, reprogram the host immune system in a way that makes it pro-reparative, anti-inflammatory, and it's now considered that these endogenous immune cells, which have been reprogrammed, are second mediators, which as circulated traffic through the bloodstream, reach the target organ and convey the protective signals. This is actually supported by now different experimental studies showing that this trapping is definitely associated with beneficial changes in the phenotype of endogenous immune cells, which are associated with improved functional outcomes. So to summarize, you see that we started with a simple concept of remuscularization, which is very difficult, definitely, to achieve. Then we move to paracrine signaling. But currently, the prevailing hypothesis is rather immunomodulation. Just to finish, this is a brief summary of the extracellular-based clinical trials that have been completed so far. And you see that you have a bunch of trials currently ongoing, spanning a very wide variety of diseases. And the last one is the one that we're going now to start very, very soon. That said, I would like once again to thank you, but my final slides maybe are the most important because I'd like to acknowledge all those who really did the work. Obviously, my team in the lab, as you can see, I'm a lucky man. I'm a rather lucky man. But also, our colleagues in the US who prepared, in particular, Nisan, who prepared the cardiovascular and progenitor cells, and obviously my colleagues in Paris of the JMP platform responsible for the translational steps of this one. Thank you very much.
Thank you, Dr. Philip, for this interesting talk and very interesting topic regarding the cardiac repair. Just, I will have a quick question before giving the questions for the attendees. Compared to hydrogels, the use of hydrogels, the EVs as advantages and disadvantages compared to hydrogel, it seems better results to hydrogels in general?
Yes, absolutely. Absolutely, because it lengthens the time of exposure of therapeutics to the myocardial tissue. If we just inject, the EVs or even the cells, they're really flushed out very rapidly. If they are incorporated into an hydrogel, they will release over time, let's say over a couple of days, and obviously this will optimize their therapeutic benefits.
Because they die quicker, I think, in hydrogels compared to EVs.
Because
They die quicker. In hydrogel, their survival is less.
well, they survive, but more importantly, they are retained in the tissue for a longer period of time.
Okay. If you have any questions? Yes?
I don't know if I can have...
Yes.
Great talk, Philippe, as usual. So I have one question because you know that the production of the extracellular vesicles needs some stress. I mean, when you culture the cells, you need to put some stress, and sometimes there's a deprivation of serum, etc., to increase the production of the LPs. So in your production process, did you have this step of stress, or? Yeah. Okay.
Yes. I did not go into too many details, but this is an important question. What we did actually, once the cells were thawed, was to culture them for a few days under normal conditions, and then to stress them with a special medium. And this is important both for what you say, that is, for increasing the production of vesicles, but also it's important because then you're sure that the medium that you collect comes from cells and not from the additives of the medium. So yes, for a couple of days they were stressed, both to increase the production of vesicles and to be sure that what we were collecting really came from the cells.
Thank you.
Additional questions, Dr. Hano?
Thank you for the talk. Well, did you explore the composition of your EVs?
The what?
Composition, the biochemical composition. Are they homogeneous or heterogeneous in their composition?
Okay, so two questions in your question. Yes, definitely we did, and obviously this was a requirement of a regulatory agency to know the composition of the cargo. So yes, we have a full description of the contents of the EVs in terms of proteins, in terms of microRNAs, so we know exactly what is inside. Now number two, maybe the slides went up rapidly, but we had two different batches of cardiovascular progenitor cells coming from the same lot. Reason was, number one, that cells being shipped overseas, we wanted to be sure there would be no problem with the customs, and in case something wrong happens, we were concerned not to lose, the whole batch. So we had 2 batches, and the second reason was that because we had 2 batches, we could compare the reproducibility of one versus the other, and they were perfectly reproducible.
Thank you for the talk. Just did you try to modify the surface of the vesicle, pegylated or something like that before the intravenous injection?
That's a great suggestion. We are currently working on that, but in the lab. For the clinical study, we start with, I would say, standard vesicles, not to add too many layers of complexity. But you're right. If you inject the cells or the vesicles intravenously, you will probably get better results if you can target them more specifically to the heart by engineering the surface or by engineering parent cells. So this is something we're currently exploring in the lab, not yet in the clinics. It's more complicated.
Thank you, Philippe. Just one small addition to the question of my colleagues. So the contents is almost the same. What about the concentration of this microRNA? Because a small difference in the amount of this microRNA may make lots of difference in the output of the right?
Well, the concentration of microRNAs are very different depending on the microRNAs because we have several hundreds of them, okay? So we were able to identify, say, 15 which were upregulated, but on the other hand, as you know, although people initially believed that there was an horizontal transfer of microRNA from the vesicles to the recipient cells, it's known that actually the number of copies per vesicle is very small. And so the increase in mRNA that you can get in the recipient cells may come from endogenous microRNAs that have been stimulated by the vesicle cargo, not necessarily by the microRNA which were present in the vesicles.
Thank you, Dr. Philippe.
Thank
Okay, I am glad to welcome Professor Roberto Accola. Professor Accola obtained his MD and PhD degrees from the University of La Spezia, Roma. His major research interest is focused on the basic mechanism of immune response against cancer cells and the interaction between a host cell and human pathogenic virus. Professor Accola presents a novel immunotherapy approach to treat glioblastoma.
Thank you very much. First of all, let me thank deeply the rector, the president of this university, Bassam Badran, who, besides being the rector, is a fantastic friend of mine since many, many years. With him, we have shared experience in science, experience in food, and experience in travel. And it's always been, for me, a sort of model, because he's a fantastic biochemist and made important advances in science. So for me, it's a great pleasure and, again, honor to participate again to this Biobeirut, which I'm an aficionado, in fact, because I've participated to many parts of them. Okay, so thank you again. What I will talk about this morning is one of the new aspects that we are dealing with in the lab, following our tradition on tumor immunology. And the new aspect is to study what we can do with one of the most dangerous and malignant tumor of the central nervous system, which is the glioblastoma. Just to introduce the issue, I would like to refresh your mind on what the immune system does, and especially what the adaptive immune system does. In order to have an antibody response or a cellular response, you need can I? Okay, here you see the arrow? No? Okay. Okay, thank you. So, the commencement of immune response starts with these cells, which is called antigen-presenting cell. This can be a macrophage, can be a B cell, can be a dendritic cell, essentially. So, the characteristic of this antigen-presenting cell is that it exposes on the cell surface peptide antigens presented by structure which are the major histocompatibility class II molecules. Now, these class II molecules come from genes which are regulated by a single transcriptional factor called C2TA, or class II transactivator. This factor was discovered by us many, many years ago, when most of you were still not born. It was in 1983. Okay? So, following this route, the antigen can be presented to these cells, which is the basic, the most important cells of the immune system, the T helper cell, the CD4 T helper cells. And once these CD4 T helper cells are activated, then you can have downstream the activation of B cells to produce antibody and cytotoxic lymphocyte to do cytolytic activity. So you need this one, and you need this one as a major partner of the immune system. So the crucial molecule is this one, C2TA, because if you don't have C2TA, you don't have expression of MHC class II, you don't have antigen presentation. So this is the baseline. Now, what is our approach? Our approach is totally unusual, I would say, because what we are trying to do is to in a way, try to displace the tumor cell from its role, make the tumor cell, by transfecting C2TA, make the tumor cells a sort of antigen-presenting cell. So if you transfect, for example, C2TA, these cells start to express MHC class II molecules, and these MHC class II molecules, in theory, and you'll see also in practice, would present tumor antigen from inside the cell, so their own tumor antigen. And now, if they present their own tumor antigens, they can attract helper T cells, these helper CTL in this case, and the CTL can kill the tumor. So the idea is that the tumor cells should become an antigen-presenting cells of their own tumor antigens. Okay? Is this possible? Is this experimentally demonstrated? And the answer is yes. The project, our project started 20 years ago, actually. And we have verified that C2TA transfected cells express MHC class II in corresponding molecules, that these C2TA tumor cells of distinct histotype and distinct genetic background are rejected in vivo in the mice, and then acquire immunological memory. These mice, which are transplanted with tumors containing C2TA, reject the tumor, but then if you come with a parental tumor, they can reject also the parental tumor. And we have shown that this is due to an immunological memory directed by T helper cells. So you can transfer directly T helper cells and have the same effect. Now, this is the proof of what I'm saying, coming from relatively old experiments that we did when we first started the project. If you take this tumor cell line, which is a breast carcinoma on the mouse, and you transfect with C2TA, you see you go from a non-expressing cells to a very big expressing cells in terms of plasmorphins. And here, this is the crucial experiment. If you put now these tumor cells in vivo, the parental cell, not transfected, will grow very fast and kill the mouse, whereas the transfected cells are rejected and the mice are alive. And this has been reproduced in many tumor systems. Now I show you one of the crucial experiments. This is one of the best experiments we have performed in the last 10 years. And this is new. The first author of the paper here, you see, Nassereddin, Farhad Nassereddin, is a PhD student, came into my lab, sent by Bassam. And to them, and to this PhD student, and to other PhD students that are coming from Lebanon, we have done most of the basic experiments that have been done in the lab. So what is this experiment? This experiment is to prove in vivo that the antigen-presenting cells are the tumor cells and not dendritic cells or other things that you can have around the tumor. What is this experiment? This experiment takes advantage of a particular strain of mouse, which is a transgenic mouse, which has the promoter, is a sensitive activating promoter of the CD11C molecule, which is expressed in dendritic cells. This promoter contains a drug-sensitive sequence, and if you put this in this strain of mice, if you put a drug after the mouse has been treated, you can kill completely all the dendritic cells of the mouse for a given period of time. Okay, so as you see here, this is the normal mouse with this number of dendritic cells. When you treat this mouse, the dendritic cell disappeared. And now in these mice treated with this drug, you transfer the tumor, the C2TA tumor. And as you see, the C2TA tumor is rejected in these mice, even if these mice don't have dendritic cells. That has been the proof in vivo that the real antigen-presenting cell to naive mice are indeed the tumor cells treated with C2TA, those cells which express us. Okay. Now, the glioblastoma. Why the glioblastoma? Because the glioblastoma is the most malignant tumor of the central nervous system, as I said, accounts for 70% of all malignant gliomas in the population, and this incidence is increasing. It's increasing in Europe, it's increasing in the States, and it's becoming a sort of very dangerous tumor because it killed the median survival is 11, 12 months, 15 months at the maximum. And there is no, and with respect to other tumors, there is no at present there is no therapy. You can do whatever you want, but there is a recurrence of the tumor, and the tumor kills the patient. And in fact, you see here there is a disproportional mortality, because even if the tumor is 1.4% of all cancer, 2.9% of all cancer-related deaths. So there is no way that you can, unfortunately, escape death when you get glioblastoma. So it's a real important. So the present standard therapy is based on surgery, chemotherapy, and radiotherapy, and as I said, it's very, very poor results. More recently, has been applied some sort of approach of immunotherapy approach, especially based on immune checkpoint and peptide vaccination, again with good results. And the major additional difficulty compared to all tumors that are around here is what is called the blood-brain barrier. So it's very difficult to treat tumors in the brain because you cannot reach sufficient quantity of drug by injecting these patients or by treating these patients. So these are all the additional difficulties. So what we did was, the following. We took an experimental system in the mouse. It's one of the few experimental systems of glioblastoma that you have. Is the cell line, the glioblastoma cell line is called GL261. This GL261 is not expressing MHC class 2, as you see here. But if you transfect with C2TA, they express a lot of class 2. So even in this tumor, the transfection with this regulatory gene induces the expression of class 2. The expression of class 2 does not alter the cell growth in vitro of the tumor because compared to the parental cells, you see the growth rate is the same. So there is no alteration in cell growth in vitro. So what we did was to orthotopically transfer this tumor into the brain of the mouse and see what happened, okay? So from one side we transfer the parental tumor, and from the other side we transfer mice with the GL261 C2TA modified tumor. And after 21 days we look at the mice and see what they had. Do they develop tumor compared to the parental tumor? If they do develop, they do develop with the same rate, or they do develop at all? And the answer was very, very interesting, because here I draw your attention on this particular comparison. Here is the big tumor of the parental mouse, of the mouse injected with the parental cells, with the non-transfected cells. And here is the remnant of a tumor injected with GL260 C2TA. So even in the mouse brain, it seems to work, the approach. The tumor is rejected or grows very, very slowly compared to the parental tumor. Okay? Now, we follow the kinetics of the tumor growth in vivo, in this parental tumor, in this tumor, and we found that indeed there is a strong reduction in growth compared to the parental GL261 non-transfected tumor. And you see already at 7 days, the tumor grows very fast in the control and doesn't grow at all in the mice injected with the C2TA tumors. So this is a quite dramatic result, I would say. Of course, we measured the infiltration and into the tumor, and we found that the major infiltration was due to CD4 and CD8 T cells. Here depicted, here you see, compared to the parental tumor, was very, very few inflammatory cells inside. And this is the number, we measured by number all these infiltrating cells. And you see there is a dramatic increase in infiltration of CD3 T cells, which are CD4 and CD8, and also FOXP3. I will come back to that later. Now, within this particular approach, the basic approach is, the most important question is the following. Once you have these quite dramatic reduction in tumor growth in vivo, is this due to an immunological phenomenon or not? Okay? So in order to approach this problem, we made this experiment. We inject the tumor, the C2TA modified tumor, into the mice. We waited 21 days, and then after 21 days, we injected in the other hemisphere of this mice, of these vaccinated mice, the parental tumor. And we follow the growth of the parental tumor. So in other words, are you vaccinated when you get injected with a vaccine, which in this case the vaccine is this C2T8 tumor? And the answer was dramatic, even in this case, because when you do this experiment, you find that the pre-vaccinated mice now reject completely the tumor, the parental tumor. You see, this is the control of the parental tumor, and this is what you have in one hemisphere, the C2T8 tumor, and the other hemisphere, this parental tumor in vaccinated mice, which doesn't grow. So this is the immunological proof that you get memory when you do this experiment. Now, I want to go fast and show you what is now the approach we are making in the human system. Okay. So just to make a sort of resume of what I said before, what I said until now, when you modify the tumor with C2TA, you change the microenvironment of the tumor, and you transform what is called a rat cold tumor, because it's not infiltrated by lymphocytes and neurocytes, to a hot tumor which can now be rejected. Now, what is the basic phenomenon under all this story that I'm telling you? The basic phenomenon is the following. If you have an immune response in a system like this one, the immune response should be dictated by recognition by CD4 T cells and CD8 T cells of an antigen. And the antigens should be presented by MHC class II molecules, which are now newly expressed in this system. So if this is true, we can have some tool to pick up these antigens and analyze their sequence, their nature, where they are coming from. And in order to do that, we have approached in a very recent study this protein, we go fast. This question by doing a very complex approach, but relatively modern, which is called mass spectrometry. So what we do is to take stably transfected cells, purify the molecules after transfection of CD2. These are human cells now, human glioblastoma. Purify the MHC class II molecules after transfection with C2TA, and then analyze the immunopeptidome, the antigens that are expressed on these MHC class II molecules. And then we can study also the antigen presentation machinery that C2TA evoke after transfection. So it's an approach to, in fact, to purify the tumor antigens. And this is what you get in terms of shotgun proteomic analysis. You see, when you transfect the cell, the glioblastoma, these are 3 glioblastoma cell lines from human, and you transfect with C2TA, you now have an increased amount of gene expression in these glioblastoma cells. And this gene expression is related to the whole presentation machine. So you inject, you activate class II, HLA, DR, DP, DQ, but you also activate other important genes in glioblastoma that are part of the antigen presentation machinery. For example, the invariant chain. For example, the DO, DP molecules, which are accessory molecules for presentation. And when you now peel out the peptides that are expressed on this molecule, these newly expressed molecules, you see that, you see here that you can have an enormous amount of newly expressed peptides on the surface of these glioblastoma cells. And these peptides, in terms of number, you see what we have? We have identified 32,000 unique HLA class II bound peptides in these. Tell me when I'm finished. Okay, tell me when just a few minutes again. And from these peptides, we identified 279 unique HLA class II peptides which have been described for tumor antigens. So it's a huge amount of peptides. And what is most important in all this story is that through different glioblastoma cell lines of different HLA haplotype, myself, yourself, whatever, we have identified 8 proteins common to all of us, from which tumor peptides are derived and expressed on these glioblastoma cells. So this for us is very important because it can give us the idea that by following this approach we can now purify, by using C2TA, which can now render these cells expressing class II, we can now purify peptides which may have importance in terms of vaccination for glioblastoma. And just to finish up the story, I show you the sort of repertoire of genes and antigens that you are activating in the glioblastoma when you do this sort of approach by transfecting C2TA, and comparing to the real immune cells, like for example B cells. These are 2 B cells from human, and you see these are the different compartments that are presented in B cells in terms of different family of proteins that are presented in B cells, which are antigen presenting cells by nature. And when you transfect the glioblastoma cell lines of different haplotypes with C2TA, you see the picture, they are comparable. In other words, you transform a glioblastoma cells in a sort of B cell when you do this sort of antigen of gene transfer. The same is true. So, to make the story short, what we are doing now, what we believe we're doing, is by using C2TA modified tumor cells, we enrich for those protein antigens which are supposed tumor antigens inside the tumor cells, which can now be seen in these cells because the expression of class II render this tumor antigen visible by these cells. And these activate a larger number of CD4 cells specific for the tumor. Guru will describe in detail because we have collaborated and he was the director of this project. It's been applied already in human clinical trial for the hepatocarcinoma, but now we are trying to apply a similar approach to glioblastoma. And it's the following: take the tumor, the patient with tumor, isolating the glioblastoma cells, transfect them or modify them by expressing class II molecules by C2TA, elute the C2TA, the class II molecules, elute the antigens, prepare the peptide vaccine, and then injecting back to the patient in order to produce what is called a therapeutic anti-tumor vaccine. This is to make show the people who did the work, and among these people you will recognize a fantastic girl that we had as PhD students in the lab, Elise Ramia from Beirut, Farah Edin from Beirut, and this is the group now working on glioblastoma, in which you recognize Mariam Shalak from Beirut. She is the most clever girl I ever had in my lab in terms of research and production of data. Thank you very much.
Thank you, Professor Akola, for this very interesting topic or presentation as usual. So, any question?
Thank you, Doctor, for your excellent presentation. I have a question, please. What chances a product like temozolomide being an old alkylating agent, maybe since 1980s, to stay in the treatment protocol in line with those updates, like immunotherapy treatment and either vaccination?
You're asking whether we are ready to vaccinate these people?
No, I'm asking about the chances of product like temozolomide to stay in the treatment protocol being an old alkylating agent.
Well, we are in a very preliminary step. So, in terms of treatment, as I told you, in terms of glioblastoma treatment, temozolomide is the one and maybe the only drug that we have to treat this patient. But even with the best results with temozolomide, the recurrence in patients is more than 50% after 1 year, and more than 75% after 3 years. There are only 5% of patients with glioblastoma under treatment that reach 5 years of life. So at present, really, these are of course the candidates for inducing, for being treated with a possible vaccine that we are preparing. But the story is the following: no single treatment will cure cancer. Now is almost sure that you will need a different approach together to cure cancer. So radiotherapy will be one, temozolomide will be one, vaccination will be one. Now, the problem for immunologists like me is what kind of vaccination, what kind of treatment you can apply. People have tried, there have been 2 major, very big trials for glioblastoma therapy with checkpoint inhibitors, and they were interrupted on the way because they were producing no results. So checkpoint inhibitors that have been valuable, very valuable, in other cancers like melanoma, like non-small cell carcinoma of the lung, like now in kidney carcinoma, in glioblastoma don't work. So we hope that a different approach like this one that we are preparing will work.
So, thank you for the nice presentation. I have a comment and a question. First comment is that in glioblastoma, our therapy modality is still weak. We are not having a lot of drugs. It's the tumor where the people that are dying very fast. When you diagnose a glioblastoma in 5 to 6 months, it's over for the patient. And this is very nice if it will work with your studies. We used to do the same treatment for kidney cancers. We used to do a nephrectomy. We take a specimen, we take a tumor, and we will modify it. We'll take the antigenity of the tumor, and we will inject it to the patient itself, like a vaccine for the tumor. But it didn't work.
What kind of cells, what kind of modification you did in the tumor?
Only the attenuation. This they have done it in now. I'm sorry.
What?
Yeah, like you take the antigenity of the cells. It didn't expand. It didn't spread.
You know, let me make a comment on your comment. Most of the so-called tumor immunologists, oncologists, that have been dealing with tumors by immunological approaches since 20 years, well actually 30 years from now, have used the approach, which is considerable, the approach of stimulating CTL, because CTL are considered the final effectors of anti-tumor immunity. If you want to get out the tumor, you have to kill the tumor, and with the cells that kill the tumor are the CTL. So they've been concentrating for 30 years on activating the CTL, by the tumor antigens which are seen by the CTL. Completely forgotten the fact that the CTL, even if you have CTL anti-tumor, even if you have them in the body of it, don't act and don't proliferate if you don't have T alpha cells. Okay? So let me make a comment which is a rather nasty comment. but most of the so-called tumor immunologists didn't know immunology. So if you want to produce an antibody response, a CTL response, you have to have CD40 cells there. And you have to have them in good number, and you have to have them in a good activation state. Because only the T helper cells that are activated can maintain the proliferation of CTL, which are the final effector of antitumor immunity. This is a big issue that has been now appreciated by many scientists, fortunately, and now is all people who are doing tumor immunology now are focused on CD4 and not only on CD8. That's why most of the immunological approaches until now have been failed.
Thank you.
Can I? Thank you. Dr. Akola, it's as always very, very interesting. I have several questions. I will start with the first one. In your mouse model, you showed that the rejection of the transfected tumor cells by naive mice, in naive mice. So did you do the experiment as a treatment of...
Yes, that's a very good
Does
question.
it work?
This is the most important question, and I didn't talk about because, of course, the results are not so... No, let me put it that way. In colon carcinoma and in breast carcinoma models, what we can do is to treat established tumors within a certain range. So, but if you take tumors that have the parental tumor growing, you can inject C2TA tumors, for example, after 2 weeks, and have an effect on the parental tumor. But doesn't work on all tumors. It doesn't work on all tumors. In glioblastoma, it works, fortunately, because we are now doing an experiment in which we injected the parental tumor in one hemisphere, not in vaccinated mice, but in normal mice, naive mice. We have injected in one hemisphere, and then by kinetics, we followed what happened to that tumor when we inject in the other hemisphere the vaccine, the C2TA tumor. And it looks like that the parental tumor grows much slower, much slower, until 7 days. After 7 days, if you inject after 7 days the C2TA tumor, you don't have any effect on the parental tumor. So, and this is, again, is reminiscent of immunology. Because when you do antigen stimulation or vaccine stimulation as a general approach, you need at least 4 days to produce antigen capturing by macrophages or dendritic cells, processing of their antigen, presentation to T cells, and activation of T cells. So the critical period is between 1 to 4 days. So if you do that before 4 days, it works. If you do it after 4 days, the tumor grows so fast that even if you have a good response, doesn't cope with the tumor.
Unfortunately, it will not be the case with patients.
Unless
you give a preventive vaccine?
Unless you do, well, of course I didn't show that because otherwise we go too much on. Unless you treat the tumor in vivo with different approaches, which are always based on C2TA anyway. So what we are doing now is to use, and it works, we have some, it works, let's say that it works. We have some evidence on the breast carcinoma that may work. Maybe, the approach is the following. We took a virus, which is a vector virus, it's an adenovirus, and it's the only virus at present which can accommodate this big gene of C2TA. Okay? Other viruses, for other reasons, cannot. So this IDD5 adenoviruses can accommodate the entire gene of C2TA. And we have shown that if you produce this vector and you introduce this vector in vivo, you have an effect on antigen presentation. So we have injected this adenovirus modified C2TA into the tumor, and we have demonstrated that the tumor expressed class So that could be one of the approach that we are following to treat in vivo tumor-bearing animal patients.
Thank you, Roberto. Of course, the last idea is very nice idea. Roberto, I saw that you have Farah Munasruddin, Maria, Elise, and Abdullah, I forget her first name. So you end up from 32,000 peptide-associated, human-associated genes, peptide to 8, right? So you need now...
To 8 proteins. No.
8 proteins.
Yes.
So
300
you need,
peptides.
so you need to prepare the mix and all this. For this, we do have lots of students, you see. So we can get an additional student.
Yes.
For the PhD.
Will be always a great
The second point is a definition sheet, this one?
So, this is a definition, yeah.
Okay, so...
It's at the museum.
So,
this is your
it's
museum.
very nice slide.
Yeah.
Thank you, Roberto, for your presentation.
I'm pleased to introduce now Dr. Luigi Bonaguro, who received his medical degree and his microbiology and virology specialty degrees from the University of Naples, Italy, Federico II. After his postdoctoral training at the Laboratory of Tumor Cell Biology at the National Cancer Institute of Health in Belgium, he joined the lab of viral oncology at the National Institute at the National Cancer Institute in Naples, Italy, where he currently is the head. Dr. Bonaguro is a major contributor in the literature on the development and evaluation of virus-like particles as vaccine approach to HIV. He is currently coordinating the FP7 funded project, Cancer Vaccine Development for Hepatocellular Carcinoma, HEPAVAC, as well as the TransCan2 funded project, Mutated Neoantigens in Hepatocellular Carcinoma.
Thank you very much for the presentation. Before starting, obviously, I want to thank the rector, the friend, Bassam, and obviously Eli for all the organization. This is a great privilege and a great honor to be here with you today. Unfortunately, I would have come more often, but several situations prevented me from being here often. But it's nice to be back to Lebanon, to Beirut. So today I will update you on the strategies that we are currently pursuing in order to develop a liver cancer vaccine, a therapeutic cancer vaccine for liver cancer. So just to give you a few details about liver cancer. Liver cancer, if we look worldwide, we see that it's your tongue. It's better than mine. Okay. So, liver cancer is the 5th cancer by incidence overall. But if we look at the mortality, actually is the 3rd in the world, and even 2nd if we split the colorectal cancer. So it's a huge medical need all over the world. And here it's shown even better that is the 2nd or the 3rd most common cause of death by cancer in the world. So what are the risk factors for liver cancer? The 2 main risk factors are the chronic infection by hepatitis B and hepatitis C. But the other risk factors may play a major role. And this is obviously the alcohol abuse, which I think that you don't have in this country. Dietary toxins, because food can be contaminated by toxins. And there are also genetic predispositions. So again, these are the risk factors, and it's interesting that in different areas of the world, the main risk factors may play a different role in terms of percentage, but overall, the major risk factors are chronic infection by hepatitis B and hepatitis C, which are transmitted by parental transmission. So I took this slide just from a meta-analysis published in 2020, just to make a parallel between Lebanon and Italy in terms of incidence rate, mortality rate, and risk factors. And as you may see here, actually liver cancer is not a major, one of the most predominant cancer in Lebanon. And again, is confirmed that the two main risk factors are HPV and HCV, with a very slight increase between 1990 and 2017 for the risk related to NASH, so to the syndrome, to the metabolic syndrome. Completely different situation is in Italy, and especially if we look at southern Italy, because in southern Italy, for some reasons, we have a very high prevalence and incidence of chronic infection by the two main hepatitis viruses. So you see that both in men and in female, we have a much higher incidence and mortality. So what is the problem with the liver cancer? Unfortunately, we do not have a very efficient treatment strategy for liver cancer. And in fact, if we look at the very early stages of liver cancer, we may have a very efficient treatment with a high survival rate at 5 years. But, I'm sorry. Okay, but if the disease continues to progress, unfortunately the treatments are not efficient anymore, and the survival rate drops dramatically. So we need to find something which must be more efficient than what we have today. And in fact, if we make just a quick comparison between liver cancer and melanoma, you see that along the years, while for melanoma the survival rate now has a plateau because of the introduction in the treatment of the immunocheckpoint, of the immunotherapy, which is very effective in melanoma. This is not true for liver cancer. So that's why a few years ago, about 10 years ago, we entered the field and we proposed to develop a therapeutic cancer vaccine for liver cancer. Also because the number of cancer vaccines for liver cancer are very few. And in fact, if I would put here a list of clinical trials with cancer vaccine for example, melanoma, 3 slides would not be sufficient. Instead, for liver cancer, 1 is more than sufficient. And unfortunately, these trials, they all reached the phase 2 stages. None of them gave a sufficient result to move forward to phase 3 trial. This is one of the most predominant problems with the liver cancer. The liver tumor microenvironment. The liver tumor microenvironment is very immunosuppressive because we have so many cells in the tumor which are immunosuppressive, and they make very difficult for the T cells to enter the liver tumor and to be effective in terms of containing the tumor growth. So we must work on this, not only to identify specific antigens which can be addressed, which can be used for developing a vaccine, but we need also to make combinations between the vaccine and drugs which may limit the immunosuppression of the tumor microenvironment in the liver. Well, just a few words about how a vaccine works, and I'm glad that Professor Accolla gave a very straightforward background. So basically, what we do with vaccine is that we identify antigens, both class 1 and class 2, in order to elicit both CD8 positive T cells and CD4 positive T cells. And the vaccine actually has one effect: to establish the memory. Only if the vaccine is able to establish the memory, whenever we encounter the real problem, either a virus, a pathogen, or the tumor, the memory immunity can be boosted, can be recalled, and the effector T cells can be effective. We can use two types of antigens to make a tumor, an anti-tumor vaccine. We can use the tumor-specific antigens, which are basically mutated antigens, or viral antigens, such as for the liver, the HBV, the hepatitis B virus, or the HPV, the papillomavirus, or we can use mutated antigens. But the limitation of the mutated antigens is that they are unique for each individual. That's why we call it private. And it's not only unique for each individual, but they can mutate, they can change during tumor progression. So this means that although the TSA, the tumor-specific antigens, are actually the best antigen that we could use to make a vaccine, it's very limited. The efficacy, and especially, is not useful, is not feasible to use for other than the single patient in which we found that. So that's why we are more focused on tumor-associated antigens. The tumor-associated antigens are wild-type antigens, so they are not mutated, and they are overexpressed in tumor cells compared to normal cells. The benefit is that we can use tumor-associated antigens as off-the-shelf vaccine, so meaning that we can use it in all patients affected by the same cancer. But the drawback is that tumor-associated antigens are self-antigens, which may be expressed at very low level also on normal cells. So they may suffer from immunological tolerance, and that's why we need to make all we can do to break the tolerance and have the immune system to react against something which the immune system should not react against, because it's self. So that's what I explained you already, and that's the concept. So the tumor-associated antigens are normal antigens. You see the blue overexpressed in tumor cells. The tumor-specific antigens instead are different from antigens derived from normal. But I told you the advantages and the drawbacks. So what do we do in our lab? So we try to identify specific antigens. We try to make combinatorial strategies, as I told you, to combine vaccine and immunomodulatory drugs. We try to improve the immunogenicity. As I told you, the tumor-associated antigens are reflex clinical trials. So this was the study, the project that Dr. Accolla mentioned before. So this was a project funded by the European Union, HEPAVAC, and in this project we identified a bunch of unique peptides expressed in liver cancer, and then obviously going through several steps of confirmation, we ended up with our vaccine, with our pepus, or even eliminate all the Tregs in order to eliminate all the immune suppression, and then we had 9 cycles of vaccination. We screened more than 80 patients, but at the end of the day, we had 22 patients who started the treatment, and 20 of them could end the treatment up to the end of the strategy. So this was published in 2022. Obviously this was a phase 1 clinical trial, and as you know, the phase 1 clinical trial is able to give us only initial answers, namely the presence or not of adverse effects, and we showed that we had some adverse effects, but they were very minor. We had only 2 grade 4, which did not need any hospitalization, so it could be solved by treatment. So this was safe, basically. And then we showed that we could elicit immune reaction against 50% of the antigens that we had in the peptide mix. And then we were also able to show that the cells that we elicited during the vaccination were, they had several phenotypes, but mostly the central memory phenotype was increasing during the treatment. So the vaccine, as I said, is safe, is significantly immunogenic, but obviously we must, we should, and we can do better. And indeed, we have now designed a new protocol in which we keep the same peptide mix, so the vaccine is the same, the antigens are the same, but now we are combining with a different adjuvant, namely the montanide, which is very used in clinical trials for cancer vaccines, and we are adding a checkpoint inhibitor. Namely, we are adding the durvalumab, which is an anti-PD-L1, which is produced by AstraZeneca. So the protocol has been submitted to the Italian regulatory agency. It has been approved. And so we are going to start very soon this new clinical trial, HEPAVAC 201, which is again a phase 1, because obviously we have changed the formulation. So this is a new phase 1, but will be done in Italy at my institute in Bolsa. And this is just to show that, again, we are introducing the montanide and the durvalumab. This is the protocol, basically is identical to HEPAVAC 101. So again, patients will be treated, and when they are tumor-free, they can be vaccinated in combination with durvalumab. And the first patient should be enrolled at the end of May, so very, very soon. As I said, we try to improve the efficacy of cancer vaccine, obviously in the preclinical models, combining the vaccine and immunoregulatory drugs, and these are the papers that we have been publishing in the last years. But I want to finish my presentation with this new concept that we are developing now in our lab. So basically, as I told you, when we deal with the tumor-associated antigens, we deal with the self-antigens. which suffer from immunotolerance, because our immune system should not react against our own antigens, otherwise we would not be here since the beginning of the history. So when we deal with the tumor-associated antigens, we need to modify a little bit these antigens in order to make them more immunogenic. And when I say modify, obviously we have to modify in the residues of the peptides which are seen by the TCR, the T-cell receptor. Because as Professor Acolla showed you, peptides are seen by T cells only when they are bound to the HLA molecule. But in the sequence, which is about 8 to 10 amino acids, some of the residues are needed to bind to the HLA, and so they are named anchor residues. The other residues are instead looking at the TCR. So if we want to make some changes in these epitopes, we need to change something in the residues facing to the TCR. But all these can work because the TCR can cross-react. Indeed, the original theory was one clonotype, one peptide, one epitope, meaning that each TCR could recognize only a single epitope. But if this theory would have been true, we would have been blind to millions of epitopes, because there are about 10 to 15 potential epitopes that we are exposed to, but in our body we have only 10 to 12 T cells. But even better, or even worse, we have only 10 to 8 naive T cells. And given that the TCR, once made, cannot be modified, it would have mean that we would have 10 to the 7 antigens that we could not recognize. So this would have been the end of the story from the beginning. So the one clonotype, one specificity paradigm is wrong. And indeed, we have a T-cell cross-reactivity. And each TCR, a single TCR, can recognize at least 10 to the 6 different MHC-bound peptides. So there is a huge cross-reactivity. So this means that if the peptides are slightly different in the residues facing the TCR, they can be cross-reacting with a single TCR. And this is just a very nice review in 2002 showing that if this is the interaction between TCR and the peptide bound to MHC, in the molecular mimicry, and the molecular mimicry means that an antigen from a pathogen is mimicking an antigen from cells. So basically you have exactly the same interaction. Obviously we may have alternative recognition, and we may have also T cells with different TCRs on the same cell, different TCR recognizing different antigens. So this means that, or this is suggesting that the T cell cross-reactivity is very wide, is very broad. So that's why I came, together with a colleague of mine, a couple of years ago, we came out with this theory. And the theory is that the pathogens are actually our allies against cancer. Meaning that we may have an antiviral memory T cell, we may have an anti-tumor memory T cell, but actually there may be a convergence. Because, as I said, if the tumor antigen is very similar to a viral antigen, and we have been exposed to the viral antigen, and we have a memory T cell for the viral antigen, now the memory T cell for the viral antigen may become an effector anti-tumor T cell. So we have applied this to liver cancer. So beside what I showed you for the EPAVAC project in the lab, we have been doing some research in order to find additional target antigens for liver cancer. So we screened thousands of proteins, and we ended up with 9 proteins which are highly expressed in liver cancer and not in normal liver or in other normal tissues, and they are correlated with the poor survival rate in liver cancer patients. So we then predicted the epitopes for HLA-0201. We selected some of them, and this is just the list of the epitopes that we have been selecting through these prediction rounds of development. But very interestingly, and that's why I introduced the concept of the molecular mimicry, some of these antigens, some of these tumor-associated antigens identified in liver cancer, actually they are almost identical to antigens expressed by pathogens. In green, you see the identical residues in the positions of the paired peptides, but not even in identical sequence. They have the identical structure, the identical conformation. And here I'm showing in yellow, so these are the paired, these are the tumor-associated antigens, and these are the paired viral antigens. In yellow, I'm showing the contact points to the HLA facing downwards, and in pink, okay, and in blue, the contact points to the 2 chains, the alpha and beta chains of the TCR. So I think that you can agree with me that they are basically identical when you look in comparison. And this is just to give a closer view of what I'm showing. So if this is the tumor antigen, and this is the antigen derived from the virus, again, the green letters means identical residues, amino acid residues at each position of the peptide. So this is the conformation, identical. This is the peptide together with the alpha and beta chain from above. Again, very similar contact points. But if you look, if we describe in this conformation, again, yellow means contact points to the HLA, and here blue, dark blue and light blue, the contact points to the alpha and beta chain of the TCR. But even better, when we look now from above to the groove of the 2 chains of the TCR, in red we can see the footprint of the tumor-associated antigens and the paired viral antigen. You see, the red spots are the contact points of the peptides with the alpha and beta chain. Basically, they are indistinguishable. So we strongly believe that a single TCR can recognize both the tumor-associated antigen and the viral antigen. So we made, obviously, we confirmed that the paired tumor-associated antigen and viral antigen can bind the HLA, and most of all, we were able to show a cross-reactivity for the same PBNC samples with both with the paired epitopes. And so obviously we made a vaccine. We have selected tumor-associated antigens and viral antigens. We made a cocktail, and this is a new vaccine which we call IIM for our lab. So the peptides in GMP have been produced. The peptide formulation has been produced, and we think that by the end of this year we will start enrollment for this new clinical trial, and this will be the first clinical trial, to our knowledge, in which we will use viral antigens to elicit anti-tumor immunity. So finally, this is my last slide. So this is our working hypothesis. We think that if we have individuals exposed to a large number of microorganisms, I don't call them pathogens because I include also the microbiota of the gut, which obviously are not pathogens. So I call it microorganisms. So if we have individuals exposed to a large number of microorganisms, or if we have individuals exposed to a lower number of a narrow number of microorganisms, but we can give a preventive vaccine based on microorganisms that are antigens, we may end up with a very broad memory anti-microorganisms antigens. Then, if these individuals now have a tumor which exposes similar antigen, very likely the memory T cell will be promptly expanded and we will have cancer regression. On the other side, if we have instead individuals with very limited number of exposure to microorganisms, this means that the memory T cell will be very limited in terms of antigens to be recognized. Then, if the tumor starts to grow, the tumor will progress. But at this time now, we could use a therapeutic vaccine with the microorganisms. And then we will have the chance to expand these cross-reacting T cells and eventually cancer progression. So that's the end of the story. These are all the collaborators, and these are the two consortia that were funded by the European Union, and in the EPAVAC consortium, Professor Acola was one of the major players. And this is my group in Naples. Unfortunately, I don't have students or postdocs from Lebanon. These are all autochthon from Naples, but they can, they can compare. Thank you very much.
Thank you. Thank you very much, Dr. Bonaburo. We have time for 2 short questions.
I'm sorry, probably I was a little bit too long.
Oh, it's okay. It was so interesting. No problem. Are there any questions?
Thank you, Dr. Luigi, for this really beautiful presentation. And really, the interesting hypothesis of molecular mimicry is really very nice. My question is the calicivirus that displays similarity with your antigens, does it infect humans? And do you know what is the function of this virus? Because you have this molecular mimicry with the virus. So do you know?
They are human viruses.
Yes.
Well, okay. First of all, let me go back just. Let me rewind just a little bit the story. Obviously, we did not invent the molecular mimicry. The concept of molecular mimicry was established in 1964, actually the year that I was born, correlating the antigens from pathogens to autoimmune disease. So it's a very old, as I am, concept. What we now are exploring in a very deep detail is the correlation between molecular mimicry and cancer. Okay, so this was just for avoiding to take advantage of something that we did not invent. Okay, so the second, the discovery phase that we are doing is not taking into account specific viruses. So this is just a fishing, okay?
Well, my question is, do these viruses prevent actually cancer? Did you see a correlation between affected people with these viruses and less tendency to develop certain types of cancer?
Well, this is an excellent question. And when I will have the final question, I will give you to you directly before giving to anyone else. Well, I don't obviously I don't have the answer to this question. We have a proof. We have an experimental proof that I did not show today because otherwise I would have been even longer than was in HIV. Okay, so we had HIV was my first love in terms of science. HIV people living with HIV/AIDS, meaning people who have survived and live longer, they show very high prevalence of AIDS-associated cancers, which are mostly cancers associated to viruses. Okay, but some of the non-AIDS-associated cancers, such as colon cancer, breast cancer, prostate cancer, are actually less prevalent in HIV patients compared to non-HIV individuals. And we found, and we published, if you are interested, you can go into the literature, and we found antigens derived from these three cancers, colorectal, breast, and prostate, which have a high homology with HIV. So obviously our hypothesis is that the HIV, the exposure, the presentation of antigens, of HIV antigens, induced a T cell which protects these individuals from these three cancers. So this is obviously
بس خلص
must be validated. So second, this is just something that we are working on. Now we are looking for something like that in SARS-CoV-2. And I can anticipate that SARS-CoV-2 has some peptides, some epitopes for some HLA, which are very similar to tumor-associated antigens. So that's what we are currently trying to show, that either the infection or the vaccination may have established, may have induced an antiviral memory T cell, which can eventually be beneficial in the coming years.
So this is the first advantage of SARS-CoV-2, right?
Well, yes, but if our theory, if our working hypothesis is okay, we think that actually the infections are those who drive the appearance of cancer or not.
Thank you, Dr. Bonaburo.
Thank you. We can keep questions.
Okay.
Break. Sorry.
Sorry, we have to keep questions for the lunch break because Dr. Awada is waiting online. Next week.
Sorry. So, thank you, Dr. Bonaburo. We are honored by the participation of Professor Ahmad Awada in Biobeirut 10. Dr. Awada is a regular speaker in this meeting. He is an expert in this field of oncology and cancer biology. Due to an unexpected emergency, he was not able to make it. However, he recorded his presentation, and it will be displayed now. And he actually is online with us for a question after the presentation.
البداية أشكر رئيس الجامعة اللبنانية الأستاذ بسام بدران ورمداء الأساتذة بجهودهم الجبارة. حقيقة الصرح المهم لبنان الجامعة اللبنانية مفتوحة أمام الطلاب الأمين العام للدراسة، وأشكرهم على جهودهم لتنظيم الدورة العاشرة لبيو بيروت. بيسعدني أن لا أكون معكم بهذه المناسبة شخصيا بعدة ظروف، منها ظروف شخصية ومنها ظروف الوضع في لبنان، وبنتمنى النجاح للمؤتمر، وهذا بشرفني أن نقوم بهذا المؤتمر لأنه هذا المؤتمر أنا من المشاركين فيه منذ البداية. I will now move to my talk, and the proposition was to speak about the perspective in the field of oncology, in particular solid tumors. And I will focus on breast cancer as an example because speaking about all solid tumor will be a problem. So I decided to go to breast cancer as an example of what the field of oncology is moving from today and for the next, let's say, 5 to 7 years. I am Ahmed Awad. I am the head of Oncology Medicine Department at Jules Bordet Institute, and I'm a medical oncologist involved in clinical practice as well, education and clinical research. You can see here my disclosure. Let me start with this slide summarizing about what I will speak today in relation to the perspective in oncology in solid tumors, but also, as I mentioned earlier, I will focus on breast cancer. I will speak about, let's say, the progress on systemic therapy, new therapeutic concept in the management of solid tumor. Of course, here the focus will be breast cancer as an example, the specific group of patients and tumors, the perspective in relation to clinical research, the perspective in relation to public health, patient empowerment, and of course, the molecular biology advances. So I will try for all these topics, I will give some example on the perspective in the ongoing perspective and for the next 5 years. Let me start with systemic therapy in the management of solid tumors in 2023 and for the following years. As you know, in breast cancer, as an example, chemotherapy is really an important component in the therapy of the disease. Hormonal therapy for hormone receptor positive breast cancer, anti-HER2 therapy, and more recently, biological agent for a subgroup of breast cancer called triple-negative breast cancer. If I would like to focus on luminal disease, that's mean hormone receptor positive breast cancer, we'll have, in addition to aromatase inhibitor and selective estrogen receptor modulator, new endocrine agents which are now in active development, which are the selective estrogen receptor degrader. And we have already one of these cells in clinical practice. Of course, the major development in luminal disease in breast cancer, the last, let's say, 10 years was the development of CDK4/6 inhibitor, and we are now in the post-CDK4/6 inhibitor, we are developing what's called new CDK inhibitors, CDK2, CDK7 inhibitors. What is really important in the management of solid tumor, and particularly true in the management of breast cancer, the development of new kind of chemotherapy, in my opinion, called targeted therapy, targeted chemotherapy, and the ADCs, antibody drugs conjugate. And the story with antibody drug conjugate in breast cancer started with the development of anti-HER2 therapy with TDM1, and now we have another ADCs in development, in particular trastuzumab deruxtecan in HER2 disease, low expression as well high expression. So there are many ADCs in development in breast cancer, as well in HER2 disease, as well in triple negative breast cancer, but I can tell you that there are new ADCs and ADCs development other solid tumors, and this is really the future of our chemotherapy armamentarium, is targeted chemotherapy. As you know, checkpoint inhibitor under the armamentarium of the treatment of solid tumors, and that's true also for breast cancer. We know that this checkpoint inhibitor has a value in the management of patients with triple-negative breast cancer, but now we are really beyond the checkpoint inhibitor, developing other immunotherapeutic approach by bispecific antibodies, CAR-Ts, even vaccine. And so I am expecting that in the following 5 to 10 years, there are new immunotherapeutic approach will be developed. Of course, molecular target therapy with the advances of molecular biologies were developed, and we are witnessing the development of new molecular targeted therapy in the management of solid tumors, and particularly true in breast cancer. And what is also as perspective in systemic therapy in breast cancer, the development of new agent targeting homologous recombination deficiency in solid tumors, and particularly true in breast cancer as well ovarian cancer, and now we are beyond the PARP inhibitors. We are developing a new systemic therapy targeting HRD, which is called all beta inhibitor. So these are some examples of new systemic therapies in active clinical research, and we start to see some of them in clinical practice. Let's move now as perspective from systemic therapy to new therapeutic concept in the management of solid tumor, and here as a focus breast cancer. You know, with the development of liquid biopsies, of course, liquid biopsy is really a revolution in the management of the solid tumors, and my expectation is that in the following years, this liquid biopsy will help a lot to understand the disease, but also to manage the patient with solid tumor. And that will be true from the screening. So liquid biopsy will be used to screen for cancer, and we started to see some studies in this in relation to screening, for example, nasopharyngeal cancer, but also liquid biopsy, we can monitor the disease, we can monitor the efficacy of the treatment. By liquid biopsy, we can monitor the emergence of resistance to therapy. So I think liquid biopsy in the following years will be really a revolution in the management of patients with solid tumor, and particularly also and true in breast cancer. As a new concept, we witnessed the last years the development of this concept called oligometastatic disease. That means, let's say, up to 5 metastatic site lesion, and so that means this opposition to what's called bulky disease. And so this is a new concept in solid tumors, but also in breast cancer. And in my opinion, the objective to manage oligometastatic disease is to cure the patient. So today in breast cancer, when the patient develops systemic metastasis, let's say in the liver, lung, or bone, the patient will not be cured. Increase the survival, increase the disease control, we have objective responses, we improve the quality of life, but we are not killing the patient. But now by approaching this, this situation of breast cancer, we call oligometastatic disease, oligometastatic setting, and by using systemic therapy in addition to stereotactic radiation and or radiofrequency or surgery, with metastatic disease from breast cancer. Another concept which was developed in the treatment of solid tumor, and particularly true for breast cancer, the concept of de-escalation versus escalation therapy. They say the escalation therapy in order not to over-treat the patient, and escalation therapy for patients at high risk of recurrence. So the aim here is to optimize the management of patients with solid tumor, and particularly true for for breast cancer. And that's is not only in relation to systemic therapy, that's true also for radiation. As an example, for example, breast cancer radiation in the past was around 6 to 8 weeks of treatment, and now we can do radiation to the patient in 3 weeks' time and even in 1 week time. That's a de-escalation without alteration of the outcome of our patient. In the opposite, if the patient, we know the patient are high risk of recurrence, the strategy is to escalate therapy by adding a new therapy, by adding a new discipline, radiation, for example, to systemic therapy in order to control the disease. And finally, new concept who was developed in the management of solid tumor, particularly true for breast cancer, the management of what is called residual disease, for example, following neoadjuvant induction chemotherapy, and in order to adapt the treatment, the post-surgery based on the residual disease after neoadjuvant approach. Let's now move to the third perspective for the following, let's say, years, and in breast cancer. And so there are a lot of effort now to go to individualize the treatment for a specific group of patients or a specific group of tumors. Rare breast cancer, for example, secondary breast cancer, which you know this kind of tumor has normally NTRK gene fusion. This is as an example. To individualize the approach based on the, let's say, physiological age of the patient, the elderly patient, the fate and the development of oncogeriatric approach, to individualize the approach specifically in premenopausal patient with breast cancer, because in this group of patients we are sure today we are still over-treating this group of patients. And finally, as a perspective for 2023 and following year, the management of breast cancer in young patients in order to preserve the fertility. So clearly there are a lot of work done now in order to treat rare breast tumor, to manage better rare breast tumor, specific group of breast cancer like elderly, premenopausal, as well the survivorship issues following the diagnosis and the treatment of breast cancer. Let's now move to the fourth perspective in breast cancer in relation to clinical research. What's really important today, really a major problem, is to bring clinical trials and innovation. And there are a lot of new trials with new anti-cancer agents, with innovative approach. And the question today, how to move this innovation in clinical trial in the majority of patients, let's say, which meet the eligibility criteria. As you know, we are segmenting now common tumor. We have now, by molecular biology, we are segmenting common tumor. We understand better rare tumor. And so that's important that innovative approach, innovative therapies reach this patient. And so we are developing here in Jules Bordet Institute with many other centers in Europe a project called Spiderweb Project. And the aim of this project is to bring new drugs or new innovative agent to the patient in his center, in his country, or in her country, in order to offer the innovation to patient. And this project, there are a lot of hurdles to reach the success of this project, but the decision is really to move on in this direction, to really offer our patient on site in the country access to clinical trials with new agent in a rare setting as quickly as possible. Of course, digital health will help, the role of social media and network, so that will be also of help to bring this trial and new anti-cancer agent, new and innovation to the majority of patients. In relation to clinical research, the perspective today, how to reduce the administrative burden of clinical research. Today, this is really a major, let's say, hurdle to advance and to convince young generation of oncologists to do clinical research. And we are working within ESMO and outside ESMO how to reduce the administrative burden, and we published in this field the last 2-3 years. It's important also in relation to clinical research in breast cancer as well in other solid tumors to think about new models of pragmatic clinical trial, and also to develop network, let's say, which come in parallel with this revolution on innovation, molecular biology understanding, and our network, Oncodistinct network, is really going in order to develop this pragmatic clinical trial in a specific situation, taking into consideration the progress in molecular biology. Some issue in relation to public health perspective for the next year. As you know, our anti-cancer agent, our innovation are very, very costly drug called financial toxicity. That's true for countries like Lebanon and Middle East countries, but also many other countries in the world, including developed countries. We cannot continue with the same model of reimbursement of medication as we are doing today. And there are urgent need for a long-term strategy by the authorities. And we were among the first to advise the authorities to be prepared to the tsunami of new agent and innovation for our patients. And we will pity for cost reason that our patients have no access to this new therapy. So we need a new model coming from the authorities and to order to optimize the access to innovation. And the role of all the stakeholders, pharmaceutical industry, authorities, investigators, medical oncologists, and even patient play a role. And we organized here, let's say, we have an organization in Belgium called Allcan Belgium, which is part of Allcan International, looking to, let's say, to reach the efficiency in the field of oncology and to accelerate drug development from one side and mainly the access to these drugs as early as possible. Of course, big data is also an issue in relation to public health, real-world evidence, and so on. Patient empowerment is really, really important. In some countries, this is very well developed, like in the US, but much less in Europe and practically inexistent in non-developed countries. So that's really important that the patient will be involved in clinical practice and research. The drug adherence treatment is really of utmost importance. We know that more and more patients are receiving alternative therapeutic approach not proved by science and by clinical research. Patient empowerment implies the use of the digital health and patient-reported outcome for symptoms as well for toxicities, and finally, important role for supportive care. So it's really important that the patient will be involved more and more in the management of their disease, helped by, let's say, by the clinician and the oncologist and all, let's say, stakeholders of all disciplines. Finally, I will finish by something really important for you. Molecular biology understanding is moving very fast. You are aware of the hallmark of cancer. This hallmark of cancer was updated constantly. They added the epigenetic, they added the metabolism, they added the senescence to the hallmark of cancer. And that means that in the future we will have new therapies, innovation, which could target epigenetic, very complicated matter, the metabolism, the senescence. In relation to molecular biology perspective in solid tumors, in particular breast cancer, we know from the beginning that cancer cells are heterogeneous, and we know there are clinical indications of tumor heterogeneity. single-cell genomics, and that's now really, there are a lot of studies looking at this single-cell level to understand better the disease, the cancer, as well as the interaction between cancer cells. And so that will lead surely to the development of target and consequently by the chemists to new therapies and innovation. As a medical oncologist and a clinical researcher, we develop a lot of biomarker. So even if we have some success with biomarker the last, let's say, 15 to 20 years, I think we need to sit and to discuss a little bit what we achieved with the story of biomarker, because a lot of work was done, but without major success. And so, in my opinion, there are a need for a critical review of biomarker story for a new departure. With that, I would like to thank you very much. I tried to give you some perspective on the field of oncology at all levels, from the systemic therapy to patient involvement to public health to molecular biology. I focus as example on breast cancer, but what is applicable for breast cancer is applicable.
Thank you, Dr. Awada, for this very interesting presentation. Now we have time just for one or two questions.
Thank you, Dr. Ahmed. Do you heard me? Dr. Ahmed.
Yes, أنا موجود.
عم تسمعني؟
عم بسمع.
Perfect. شكرا للpresentation بتاعتك. Many thanks. Even if you are still in Belgium. Just about CDK2 inhibitor and CDK7 inhibitors. Do you think that those two inhibitors will have almost the same effect like the CDK4 and the 6 inhibitors for the HER2 negative patients, breast cancer patients?
Thanks, Dr. Bassam, for the invitation and for the question. Yes, I think we achieved what we achieved with CDK4/6 inhibitor now in the management of breast cancer. I think it was a major achievement. So now we are, let's say, looking to see other targets in the cell cycle pathway, and there are some preclinical data to support that by blocking CDK2, CDK7, for example, we will achieve more reserve, better reserve, mainly in patient resistant to CDK4/6 inhibitor. So, possibly CDK2 and CDK7 are alternative pathways which lead to resistance, and consequently, targeting this pathway could be of help to overcome the resistance.
Okay.
We are pleased to welcome now.
Thank you, Dr. Ahmed.
Thank you. We are pleased to welcome now Dr. Jean-Pierre Coury, and we thank him for his participation in this edition of Biobeirut. Dr. Coury obtained his medical doctor degree from the Faculty of Medicine of Saint Joseph University in Beirut in 2010. Then he started his fellowship in Hôtel Dieu de France, Saint Joseph University Hospital in Beirut, and he continued his medical oncology fellowship in Jules Bordet Institute in Brussels, Belgium from 2014 to 2016. He worked in the digestive oncology department in Hôpital Européen Georges Pompidou in Paris as a researcher in the Association des Gastro-Entérologues Oncologues en France. He is certified from Paris Diderot and Paris Descartes University in hereditary cancers and digestive oncology. He is practicing as hematologist oncologist in Hôtel Dieu de France. So you are welcome.
Thank you very much for this introduction. I'm honored to be with you today. It will be very difficult also to present after my mentor and my godfather, Professor Ahmed Awada. I was his student in Jules Bordet. So I will try to do my best to summarize in 20 minutes the revolution of immune checkpoint inhibitors in upper GI malignancy. So I'll be discussing now the clinical part of the immunotherapy, the big revolution that started since 2015 with the first FDA approvals of these drugs, and now it's really indicated in every malignancy. So the big part and the important part of my discussion will be dedicated for the upper GI malignancy. I specify the gastric and the esophageal cancers. So why gastric and esophageal cancer? Because they represent today around 13% of the incidence, and in this case it is really an unmet need of good treatments, because the treatments of chemotherapy stagnated for a long time in these two malignancies. Also, the number of deaths is higher than the incidence of these two diseases, and we don't have the right medication till now. The chemotherapy didn't find the right solution. So the incidence of gastric cancer in our region is different according to each country. We have the higher incidence probably in Yemen and Oman, but also we have other particularities of this gastric cancer regarding the epidemiology and the characteristics and the risk factors. What is important today in GI malignancy, and we can treat them before having clear biomarkers. The three important biomarkers are the MSI, the HER2 and PD-L1 in gastric and esophageal cancer. Without them today in 2022, we are not able to treat correctly our patients. The MSI, its importance today, it's an agnostic biomarker. We should define it in every tumor because it's an important predictive biomarker of immunotherapies. The HER2 was first described in breast cancer, where we have a targeted therapy called the trastuzumab, one of the first targeted therapy that revolutionized the management and treatment of breast cancer. And the PD-L1 is also an important biomarker for immune checkpoint inhibitors, but it depends from the tumor type, the stage of the disease, and many other factors, and is also dependent from the pathologist that will read the biopsy. So the PD-L1 is an important biomarker, but it's difficult to validate it. So three biomarkers that we will use them in discussing the pivotal clinical trials. In the advanced gastric cancer, the last approved drug was the trastuzumab in 2010 with the TOGA trial. And from 2010 to 2021, we didn't have any approvals. So this was really frustrating. In 2021, the first immune checkpoint inhibitors, the nivolumab, was approved based on Checkmate 649 results. And since then, we have many immune checkpoint inhibitors that are approved in the management of gastric and esophageal cancer. So I will try to summarize in this talk 6 of the pivotal trials that led to the FDA approval of immune checkpoint inhibitors in the esophageal cancer, in gastric cancer, and now we are talking also in early esophageal cancer. So first, the immune checkpoint inhibitors were approved in metastatic setting disease. Now we are using them earlier and earlier, and also in bladder cancer today, when we have superficial tumors, immune checkpoint inhibitors can prevent to be transformed to invasive cancer. So this line of immune checkpoint inhibitors showed a revolution in the management of cancer. So let's start with the advanced esophageal cancer with the Keynote 590. When I talk about Keynote, is pembrolizumab. When I talk about Checkmate, is nivolumab. So when I say Keynote 590, I'm talking about a trial based on pembrolizumab. So this study, it's a phase 3 study trying to compare chemotherapy and chemotherapy with pembrolizumab in advanced and metastatic